Trevor Graham aslında hepimizden biraz farklı olarak farklı bir eğitim kariyerinden başladı şu anki noktasına. Kendisi lisans eğitimini Imperial College of London'da matematik üzerine okudu. Matematiğin sonrasında şu anki Londra'da olan ünlü Crick Enstitüsü'nün bir önceki halinde doktora sorusu çalışmalarını gerçekleştirdi ve belki de bugün göreceğimiz konuşmanın temellerini attığı kanserle evrimsel prensiplerin birleştirilmesi yönündeki çalışmalarını postdoktora çalışmalarını yapmak üzere Amerika'daki UCSF'deki Carlo Mieli'nin laboratuvarında başladı. Ardından 2013 yılında Londra'ya Barts Kanser Enstitüsü'ne grup lideri olarak döndü ve 2019 yılında ise yine Londra'da yer alan ünlü Institute of Cancer Research yani Londra Kanser Enstitüsü'nde kanser ve evrim biriminin direktörü olarak görev yapmaya başladı. Thank you so much, Trevor, for being us today. So we are happy to listen to you.
Thank you, Ahmet. Thank you for the invitation, and thank you all for taking the time to listen to me. So I want to talk about something a little different. I want to talk about, once we have all of this incredible data, we have all of these whole genomes, how can we make best use of that data and learn new things about the biology of cancer and also new features of the genome that give us targets that we can exploit clinically. So as you've heard, I'm director of the Centre for the Evolution in Cancer at the Institute of Cancer Research in London in the UK. And so, as you would imagine, given my affiliation, we take an approach where we take ideas from evolution and try and apply them to make sense of genomic data and get new insights that way. The work I'm going to tell you about today was mainly funded by Cancer Research UK and by the Wellcome Trust, and a little bit from the US National Institute of Health. Okay, so what do I mean when I talk about cancer evolution? Those of you who've studied this will have a picture a bit like the one in the screen in your mind. So this is a graph. This is on the x-axis we have time, and on the y-axis we have the size of a clonal expansion. And so when we're talking about the evolution of cancer, we're talking about this process that transforms normal cells, they're the ones that are in the blue on the left-hand side of the diagram, into those dark red cells on the right-hand side of the diagram. Those dark red cells might represent metastatic or treatment-resistant disease. So the process that transforms healthy cells to metastatic treatment-resistant cells is an evolutionary process. There is an acquisition of mutations or possibly epigenetic changes in those healthy cells. Those key mutations that change cell biology in my diagram are indicated by the red cross. And then each of those mutations that's important for cancer biology will drive a clonal expansion. It will make the cell carrying those mutations produce more surviving offspring than other cells in that cancer. And so that drives these waves of colour that you see expanding across the slide. And so what we're interested in learning is, what is the dynamics of this process? How long does it take? How fast are those clonal expansions? How many there are? And perhaps most crucially, what are the genetic events or epigenetic events that drive those clonal expansions in the first place? What's the identity of those red crosses? Or I have a terrible joke, which I hope translates. We're interested in which mutations drive cancer evolution. And there's this movie that you may have seen called Drive, which stars Ryan Gosling. And I'm interested in which mutations are Ryan Gosling in the cancer genome. And so this is really what I want to talk about today, is finding Ryan Gosling in the cancer genome. I apologise for the joke. So the state of the art for doing this is to take the biggest set of data that we can possibly find, and then we break that down into all the data, say, for a particular type of cancer, like colon cancer, or a subset of lung cancer, or breast cancer, for example. And then we look across that cohort at each gene individually and look for patterns of mutations in those genes that indicate that gene as a driver. So one of the very best statistical pipelines for doing this is called Intergen. It was developed by Nuria Lopez, who's based in Barcelona. And what Intergen does is look at these three features you can see indicated in the middle of the slide. So the first feature is to ask whether or not a gene has more mutations, more non-synonymous mutations to be precise, than you expect compared to chance. So if a driver is important, it is likely to be mutated much more than a gene which is not important. Then you can ask, well, where do the mutations fall within that gene? That's the middle section of that middle diagram. And if a gene is a driver, then the mutations will tend to cluster in a functionally important part of the gene. And so KRAS, for example, is a good example of this, where nearly all of the mutations in KRAS, which are oncogenic, fall in just two amino acids, codons 12 and 13, and they constitutively activate the protein. So if you look genetically, you see that those mutations just fall in that small region of the oncogene. And then finally, the bottom row in that middle panel is if we have additional functional information from experiments in the lab, say in cell lines or in a mouse, you can integrate that information that you know. And so then if you put this all together, you can run it on a type of cancer. And so, for example, if you run it on colorectal cancer, if you go to the Intergen website and run it on colorectal cancer, you output this list of genes, and I'm sure you can read the names of those genes at the bottom of the screen, and you'll recognize many of the genes. So, of course, APC, the most frequently mutated gene in colorectal cancer, comes up as a driver, and P53, probably the most famous cancer gene mutated in many different types of cancer, is on there, and KRAS, and then there's this long tail of genes which are indicated to be driver alterations in these cancers. And so I want you to just take a step back for a moment and think about what this mutation information, this driver information actually means. What this means is that on average, across the cancers in the cohort, these genes are acting as a driver. And that's very different to saying in an individual cancer that this mutation in this gene is acting as a driver. And of course that latter part, that individual information about whether or not a mutation is a driver in a cancer, is key to personalized medicine. Because we may have a mutation in a gene which usually is a driver, but for some reason perhaps the environment of this particular cancer, the microenvironment of this particular cancer, is quite different to the average. That mutation, which is usually a driver, is not a driver in this cancer. And so if we were going to base treatment decisions on just whether or not there is a mutation in the cancer, we risk getting the answer wrong and giving a drug where it might not work, or missing the opportunity, say, to give a drug where it could work. And so what we really want is to go away from this cohort level information and have individual level information on a cancer. And that's what we've been trying to do. So it's really important, this individual level information. Here's a time course from a colorectal cancer patient that we've been studying in the lab. All of these samples are from that one individual, and they're followed for many years, as you can see from the timeline at the top. This patient is obviously very unusual. Most patients with colorectal cancer live only a few years, sadly, but this patient lived for a long time. And so you can see it just illustrates how unique individual patient trajectories might be, and how, therefore, that different driver alterations might act differently in different people. This patient, just for your knowledge, had just the classic APC, P53, and KRAS mutations in their genome. So the problem gets harder when we have more data, ironically. Usually, if you have more data, things are better. But with genomics, I'm not sure that isn't always the case. So what we've been doing recently is to sequence many spatially different regions of the same tumour to look at the heterogeneity in the genome that exists within a single tumour. So there are lots of differences between cells in the tumour. So what we do is sequence lots of individual samples. That's these, each of the tips on these phylogenetic trees you can see on the screen. And then we can reconstruct the family history of each of those samples and build these phylogenetic trees. Once we've built those phylogenetic trees, these cancer family trees, we can annotate on the tree mutations that fall in genes which we think are likely to be drivers. And so in colorectal cancer, where these data come from, most of the drivers that you can see annotated tend to be on the trunk of the tree, but there are some which are in some but not all of the samples in the cancer. And the question is, are those ones really drivers? Are they really shaping the biology of these cancers? And of course, these are in mutations in genes which we think are likely to be important, but the question is, are they really? What we find is that if we sequence more samples, we find more and more mutations. That's why the problem gets worse when we have more data. We might suddenly find lots of drivers if we sequenced 100 samples from a single cancer. And so then we have a problem understanding which ones are important. So which ones are the real drivers? Which ones are Ryan Gosling? Of course, if we could just observe this directly, it would be easy. If we could put a camera inside and watch mutations arise, and then see if they drive a clonal expansion or not in an individual cancer, we would know directly whether or not they were drivers. But for very obvious reasons, we can't do that. So the next slide is slightly gory. If anybody is squeamish, please close your eyes for a moment. So the problem is that all we get to analyze, as you well know, is at the end of the process. So this is a colorectal cancer, for anyone who has their eyes closed, a big, ugly colorectal cancer about the size of my fist. And so this is all we can analyze. But what we want to know is what's happened in the past, what happened before this sample grew and ended up in our laboratory. How did it grow? And we can't observe that directly. So if anyone is looking away, the gore is now gone. So now you can open your eyes again. So now what we do is say, well, we think this readout of how the cancer is grown is written in the pattern of heterogeneity that will exist within a single individual cancer. And so the idea is very analogous to the rings inside a tree, where you know that the width of a ring tells you how much a tree grew in a particular year. And you might think about taking a sample from the tree ring, and you would know about the environment the tree was in that year. And so much in the same way, the pattern of mutations across the cancer genome and their heterogeneity within a cancer is a readout of how that cancer grew. So to read that secret diary, to read the tree rings of the cancer genome, what we do is use mathematical modeling. We make a mathematical model that predicts what the pattern of heterogeneity will look like in a cancer if that cancer is evolving in a particular way. And so the mathematical models are illustrated on the left of the slide here, where each of those shapes which shows up is a new mutation, and you can see the cells dividing as you move from left to right in that left-hand picture. And those mutations are, of course, inherited by the daughter cells. So you can see how the cancer grows shapes the pattern of mutations that exist within the cancer. And so this heterogeneity information is naturally found, naturally generated by whole genome sequencing data. So on the right of the slide, you can see a plot of whole genome sequencing data. This is from a stomach cancer, this particular one. And what we're plotting here is something that we call the variant allele frequency, or VAF, histogram. So on the x-axis, you have the allelic frequency of a mutation. And on the y-axis, so that's equivalent to the number of cells that mutation's in. And on the y-axis, you have the number of mutations seen at that frequency. So the mutations on the right of the plot at high frequency, they're the clonal ones. They're present in every cell in that cancer. And the ones in the intermediate region of the plot, they're subclonal. They're in some but not all of the cells in the cancer. So those subclonal ones are characterizing tumor heterogeneity. And you can see from this plot that they do it in a quantitative way. We see very few mutations at very low frequency on the far left of that plot, but that's just a false finding. That's because the sensitivity of the sequencing is not good enough to see those very rare mutations. And so our question is, this pattern of heterogeneity from those intermediate frequency mutations, how is that produced by how the cancer's grown? And that's what we try and learn with our mathematical modeling. So we worked on this for many years. The first question we asked is, if cancer evolution is really boring, if all of the cells grow exactly the same and there are no drivers within that cancer, what does the pattern of heterogeneity look like? And so we have a mathematical model where every cell has the same chance of producing surviving offspring. If we plot the data there, we see a distribution like this, where there are more and more mutations at lower and lower frequency. And you can write down mathematically what this distribution is. It has a power law distribution. You can read about it if you're interested in the references on this slide. So then we can ask, well, what happens if there is a driver? What happens if some biology has changed in this cancer and driven an expansion? So in the diagram, the cartoon in the bottom left, you can see the red cell has appeared and now it grows faster than the other cells in the cancer, faster than the blue ones. So we can simulate that on the computer. And if we look at the data that we expect to get out, our pattern of mutations, what you see is this additional bump in the distribution. You see this, this extra bump here in the distribution. That bump is driven by all the mutations that have accrued before the subclonal expanded and then are carried to higher frequency as that clone is growing. And it turns out you can say very quantitative things about the selection force that driver has experienced. You can say when the mutation showed up and how fast the clone is growing by looking at the shape and frequency of that extra bump in the distribution. So you can have a quantitative test about the dynamics of the past with data just from a single point in time. So you can ask, is there a driver changing the biology of this cancer? That's a dynamical process, but you can read it out just from this snapshot at a single point in time. And I still think that is amazing. And so here's some real examples. This is a metastatic lesion that originated in the brain. This is data from the Hartwig Medical Consortium cohort in the Netherlands. You're seeing these variant allele frequency histograms here. You can see that green cluster of mutations indicating there is a subclone here. And you maybe can just about read at the top where we've annotated on the frequency of mutations in genes thought to be drivers in glioma. And you can see in the right-hand plot that there's a KMT2C mutation, a chromatin modifier mutation, in that subclone. So that mutation is almost certainly acting as a driver in this particular cancer. Whereas we may have found that that mutation was in the tail of those neutral mutations on the far left, then it wouldn't have been a driver. It's not functionally acting as a driver in this cancer. So we can have a test now for individual cancers about whether or not a mutation is a driver or not. And we might think then about being able to make personalized decisions based on this evolutionary information. So I want to spend a little bit of time talking about spatial data. So I've told you already about the heterogeneity that exists in a cancer because of the differences between cancer cells. You can visualize that heterogeneity very beautifully by staining with a mutation-specific probe on the RNA. And so that's what you're seeing here. Here are 3 different colorectal cancers where we have a mutation-specific RNA probe for that mutation written in red at the top of each picture. And what you can see is where cells are red in the image, that's where the mutation is. Where they're yellow in the image, they're cancer cells, but they're not mutated. And in the background, white color is non-cancer cells, so immune cells and fibroblasts, muscle, so on. And so you can see from these pictures that the cancers have a really beautiful clonal structure. They're very intricately patterned by these subclones. And so the maths I showed you ignores all of that. And so we can do better maths to take into account the spatial structure in a cancer. And it turns out if you have many samples, you have much better power to detect selection, but you need different computational ways to make sense of it. So what we can do is grow a tumor in space on the computer. You see 2 simulations here. On the left, that tumor's neutral. There is no driver there. On the right, there is a subclonal driver that makes the cells go red in my diagram. And on the bottom, the same simulations are recolored by all of the passenger mutations, the ones which don't drive evolution. And there you can see the clonal structure that exists in these cancers. We can simulate the process of taking multiple samples from a tumor, those black circles, and then we can simulate the process of building trees, phylogenetic trees from these multi-region data. So you see 4 trees here. The 2 on the left are from the neutral simulation, the 2 on the right from the selection simulation, and there are more or less samples on the left and right tree of those pairs. And so when you look at these, you can see straight away that the shape of the tree, when there is a driver there, when there is selection, is very different than when there is no driver. You get these long, sticky out branches when there is a driver. And so you can take the phylogenetic trees and use it as a test for whether or not there is a driver in an individual cancer. And this is a very powerful way of finding selection in cancers, much better than having a single sample, as I showed you a moment ago. So what we did is generate lots of multi-region data, slightly gory again, I apologize, from colorectal cancers. We took multiple samples, 4 around the clock from these colorectal cancers, and with each of those large samples we took many, many small ones that you can see on the slide. And then in each sample we did high-quality multi-omics. We did full transcript sequencing, we did chromatin accessibility sequencing called ATAC-seq, and we also did deep whole genomes in each sample to about 45x coverage. And we generated this huge set of data with fairly good overlap between the omics. So now if I just, and this data is publicly available, you can download it, there's a link within the papers if you want to play with it. So then what we did is take just the genetic data and we built these phylogenetic trees for all of these different cancers. Here is the forest of trees that you can see from these cases. Some people I can see are trying to spot selection on these trees by looking for those sticky out branches that I mentioned before. I think there is only one good example on this slide. It's this one here. You can see that this one has a long branch which sticks out, and you can maybe read that that subclone in the cancer has a KRAS mutation. So in this case, that KRAS subclone is a driver in this cancer. All of the other ones, it's hard to see. You would have to argue that they look different to a tree in the neutral case. So what we did is make a statistical computational method to quantitatively detect selection in these trees. If we run that, we can classify each cancer individually about whether or not it's likely to have a subclonal driver. The majority of cases, about half, we were very confident there was no subclonal driver there. Only in about 6 of the 26 where we had good data, we thought there was a subclonal driver, and the rest we weren't sure about, and so we would assume they're neutral, no driver, because that's a simpler model. So then, of course, this classification is on an individual cancer. So then we can go back into the data and ask, is there a driver in this cancer for real? There is a mutation in a gene which we think is a driver, but is it actually acting as a driver in this individual cancer? And so this is the example I highlighted to you a few slides back in that forest of trees. This is the one with the subclonal KRAS mutation, and the shape of this tree is indicative that that KRAS mutation is a subclonal driver because it changes the shape of the tree. But in these two on the right, there are subclonal mutations in genes which on average are drivers, but our computational method says they don't change the shape of the tree enough for us to think they're really acting as drivers here. And so a really interesting example is this one in the middle of the screen, where there's this subclonal mutation in PI3 kinase. I think this is druggable, but we do not think that this mutation is acting as a driver in this particular cancer. So this is where this kind of information could guide personalized medicine. So if we try and put it all together, return to the Ryan Gosling joke, where there is a subclonal driver in the cancer, where there really is a Ryan Gosling there, how often does that list of driver mutations pick it up? How often do we know there is a driver when there really is one? And the answer is about 2/3 of the time. And so that means there are about 1 in 3 drivers left to be discovered. So there is more to learn from the genome. And you can think about that as the sensitivity of our driver gene list as being about 2/3. If we take it the other way round, where someone is pretending to be Ryan Gosling, here's a picture of Harry Kane, who has a striking resemblance to Ryan Gosling, I think. So here we think the tumour has no driver in, but there's a mutation in that tumour which we might, if we didn't have this information, think was a driver. That happens in about 2/3 of cases. So we can risk overcalling drivers by a huge amount. And so this equates to the specificity of that driver gene list being only about 1/3, so being quite low. These data are just from colorectal cancer in our small cohort, but this idea, I think, should generalize across cancer types. And it will be very interesting to know how these sensitivity and specificity numbers generalize or don't. I've got 2 more slides where I can stop. Great. So on here you can see that there is 1/3 of cases where there is a driver there is a subclonal expansion, but we don't know what it is. We don't find the driver gene mutated in that case with the subclonal expansion. And so the question is, what is driving the evolution in these cases? And so I showed you before that in our cohort we have multi-omic data. One of the data types that we have, which there isn't very much of in cancer, is measuring chromatin alterations by a TAC-seq. And so what we look for is something that we've named somatic chromatin accessibility alterations, or SCARs for short. This is where the architecture of the chromatin is changed in the cancer compared to the matched normal tissue from these patients. So in some regions the DNA might be tightly folded in the normal tissue, so the genes encoded in that tightly folded DNA cannot be expressed, whereas in the cancer the accessibility might be very open, so it's unfolded and those genes can be expressed. This is data from one particular cancer in our cohort, where each of the coloured lines here shows you the chromatin accessibility from a different spatial region of that cancer. Where there are lines go up, where there's lots of read coverage, that means the chromatin is open, so it is accessible. And so if you look at the orange line, there are very few reads, the chromatin is closed at this location in the genome, whereas in the cancer there are lots of reads at that location, those peaks, and so the chromatin is open. So this gene is accessible. As you can read, this is the JAK3 promoter. JAK3 is thought to be an oncogene in some cancer types. And what we're seeing here is there's no genetic mutation, but there is a somatic change in the epigenome which can enable this gene to be expressed. And so it's possible that these epigenetic changes are themselves drivers of cancer evolution. And so if we look across our whole cohort, each of the columns in these heat maps is one particular cancer. Each row is a different locus in the genome. On the left we have promoters, on the right we have enhancers of genes. And where you see a colour, where it's red, there's a gain of accessibility, and where it's blue, there's a loss of chromatin accessibility. What we can see across cancers are that there are recurrent changes in the epigenome structure, and often those changes happen at genes which we think are likely to be oncogenes or tumour suppressor genes. And so you can read some of the gene names here, and I'm sure spot a favourite gene of yours, which may or may not be driving these cancers, but I think this is very suggestive information about a causative role for the epigenome in cancer evolution. And so I show you this to say how you can combine multi-omic data within this evolutionary framework to find new important parts of cancer biology, new drivers of cancer evolution. So to summarize, if there is a driver mutation, if there is positive selection, that changes the pattern of tumour heterogeneity in a way which can be very accurately predicted by computational models. And that means that you can work backwards from the pattern of heterogeneity you observe to what was the underlying evolutionary process of that cancer. That gives you a way for an individual cancer to understand how it has evolved and whether or not individual mutations in that cancer are important for that cancer's biology. And interestingly, when we apply this methodology in colorectal cancer, we see that there is sometimes a mismatch between what we think is a driver and what is actually a driver in a particular cancer. And it's very interesting to discuss why that mismatch can occur. And so just to thank the key people here, all of the work of the multi-omic work is done by Jacob Hausum and also by Timon Heidi, who worked between Andreas Otteriva and my labs. It's been a long-standing collaboration with my friend and colleague Andreas Otteriva, who's now based in Milan, also with Giulio Caravagna, who's now based in Trieste in Italy. And I mentioned the funding primarily comes from Cancer Research UK and from the Wellcome Trust. So thank you very much for listening.